Welcome to the Mechanisms of Developmental Patterning module. In this talk, we're going to look at the different steps by which the cell is able to finalize its fate in the organism. This includes the process of specification, determination, and differentiation. The development of an organism begins right after fertilization when that single-celled zygote is formed. Now that zygote undergoes mitosis to become a multicellular organism. Now these cells that are undergoing development have to acquire different identities in that embryo, and that depends on their relative spatial positions. This process that allows the developing cell to acquire these identities is called patterning. Now, patterning or pattern formation ensures that tissues and organs are able to develop in their correct places and orientation. Now, before the cells are able to form these different patterns, the cells must first determine their fate, and that was what helps them become a certain part of the organism's body. The different stages that are involved that enable cells to determine their fate in the body include the stage of cell commitment. Now, cell commitment requires the process of specification to occur, followed by the process of determination. Once cell commitment has been finalized, then the cell undergoes differentiation, which allows the cell to develop specialized structure elements and distinct functional properties. Now let us look at each one of these stages.
Let's start with specification. This is the first stage of commitment. Let's take an example of the blastula stage. The blastula stage occurs when the zygote undergoes many cell divisions during the cleavage process of development. Let's look at the picture, and here we have a blastula where the red cells are cells that are supposed to become muscle cells, whereas the yellow cells are the cells that will eventually become neurons. If we take one of these cells that will become a muscle cell and place it in an environment that is neutral, where the cell is not provide any kind of information of what it is supposed to become, and if the cell is able to still become a muscle cell, then specification has occurred. Similarly, if we took one of the yellow cells where the cell is supposed to become a neuron and place it in an environment that is neutral, and that cell is able to actually develop into a neuron, then the specific cell has undergone the process of specification. For cells to be committed, the first stage was specification, and the second stage of commitment was determination. A cell is determined if a cell is able to differentiate into its specified fate when it's removed from the blastula stage and transferred to part of the embryo or a cluster of differently specified cells. To understand what I just said, let's look at the figure. So let us consider a blastula where you have two different types of cells. The red colored cells are cells that are supposed to become muscle cells, or muscle tissues, whereas the yellow colored cells are the cells that are destined to become neurons. If we take one of the muscle cells, which is the pink cell, and culture it in an environment where you have the neuronal cells and observe the fate of that muscle cell, if it actually becomes muscle despite the presence of the neuronal cells, the specification is irreversible, and that cell is said to have undergone determination. However, if that muscle cell turns to develop into a neuron due to the presence of the neuronal cells around it, then that means that early cell that was chosen from the blastula did not undergo determination, and thus the specification is labile. Thus, in cells that have undergone determination, the commitment to form a specific lineage is irreversible. Once determination occurs, the next stage in development is cell differentiation. So as we saw earlier, the embryo is initially made up of undifferentiated cells that undergo commitment through the process of specification, followed by determination. After commitment occurs, the next stage is differentiation, which is a process by which the cell develops specialized structural elements and distinct functional properties. What we mean is this is the stage where a cell becomes either a muscle or a neuron, which are distinct in their structures as well as their functions. Differentiation usually occurs due to the expression of a unique combination of genes that are expressed in that cell. We're going to learn more about the differentiation process in Chapter 3. With this slide, we come to the end of Chapter 2.
With this, we come to the end of our talk, where we learned about the different steps that are involved in the cell determining its fate in the embryo. And these included the processes of specification, determination, and differentiation.